I'm sure you've heard of the term epigenetics, but do you know what exactly it is? Let me explain. Think of your genome as a massive recipe book with instructions for everything needed to sustain life. This book has around 20,000 chapters, or genes. At any given time, our cells only read certain chapters from this book to produce what they need. Other chapters are stapled shut, so they can't be read. For example, a stomach cell needs genes to make acid and not keratin, which is used to make hair and nails. Every cell in our body contains the entire genome, but each cell can only read the genes necessary for its specific function. This selective reading, which operates above the genetic code level, is known as epigenetic modification. Cells can turn genes on or off using this method. But how exactly do cells do this? One way is through histone modifications. Imagine your DNA as a long thread wrapped around tiny spools called histones. These histone proteins have long arms that can be chemically modified to wrap tightly around the DNA, silencing the gene by preventing the cell from physically accessing that gene. This silencing is crucial as stem cells grow and divide, becoming specialized cells like white blood cells, liver cells, or neurons. Each cell can only access the genes it needs for its job, keeping things orderly. This is like an orchestra playing a beautiful symphony rather than all instruments playing at the same time. Another example of epigenetic modification is through direct DNA strand modification, which happens to a lactase gene as we grow older, causing many adults to become lactose intolerant. The genetic information to produce lactase enzyme is still there, our cells just can't access it. Epigenetic modifications can also have serious implications for our health. Environmental stressors like childhood food insecurity can cause complex epigenetic changes that can lead to adult obesity, diabetes, and even heart problems. We are just learning to understand the complexity of epigenetics, and it is opening new avenues in science and medicine. We are discovering ways to reverse gene silencing, enabling the creation of stem cells from already specialized cells called induced pluripotent stem cells, or iPSCs. We've even made sperm cells out of skin cells. This breakthrough has immense potential in medical research and treatment, and I'm personally very excited to see where this goes. If you enjoy learning about optimistic science, follow for more.